Hello, my dear friends and students. A very warm welcome to again another session of a power-packed zoology part, that is Power Hour Zoology, Bio Mentor, Bio Guide, Bio Friend, Dr. Ami Bayani. Now today we are nearing towards the end of these Power Hour sessions with zoology. We are into the second last topic of Power Hour, where we are going to take an important topic, that is biotechnology, principles, processes, and biotechnology and its application. As far as NEET is concerned, my dear friends, you are all, I think, aware that these two chapters, which is Unit 9 of our NCERT Standard 12, holds a very high weightage. There are approximately 8 to 10 questions that are coming from this chapter. So practically it is 40 marks chapters, which are both of these. They are absolutely simple as we've studied the last few chapters. I hope the last few chapters are absolutely crystal clear. There is not even a single doubt about any of those important subtopics which we've covered. The same way we will be taking up these sessions and these chapters, and here also I guarantee there wouldn't be any problem in these chapters also. So stay tuned while we go ahead with these chapters. But before that, as I was saying, there are approximately 8 to 10 questions that come from these chapters. So prepare these chapters absolutely thoroughly well, because 4 marks here or there is like thousands of ranks up and down, isn't it? So let's get started. What are the topics we are going to discuss? Now we are going to deal with this chapter in a little different way, where we are going to discuss about the tools and the step in which that tool is used simultaneously, so that it becomes very, very easy for you to understand it, okay? Then we are going to go into the application into agriculture, where we are going to take one important example of BT cotton, from which large number of questions are coming, and it is repeatedly coming. And we are also going to talk about application in medicine, where we are going to see that how insulin, diabetes capital of the world is our country, that is India. So how insulin plays a very important role, and to provide insulin to every individual who needs it, how biotechnology helps in it, okay? So we will be discussing these two topics of applications here. Now let us understand what do we mean by the word biotechnology. Now, as the name suggests, biotechnology. Bios means life, and technology is techniques. Now, we've been using various techniques with the use of living organisms, with the use of its parts, with the use of its various enzymes to obtain processes or to obtain products for our own betterment, so that our lifestyle and our living becomes better and better. So how do we use this? Is this something absolutely a new field, or this has been existing since ages now? Our grandmas, their grandmas have been using this since ages without exactly knowing how beautifully living organisms are helping us to make our lives better. Say for example, you've eaten curd, right? You've eaten dosas, you've eaten doklas, you've eaten idlis, you've eaten cheese, and these you've seen people kind of, using these beverages, that is wine and beer. How are these prepared? Be it curd, be it dosas, be it cheese, be it beer, be it wine, be it alcohol, it is by the process of fermentation. Respiration chapter botany, you must have remembered fermentation, which is another term kind of synonymous with anaerobic respiration, isn't it? So here also we are using living organisms. In milk we are putting lactobacillus, again your microbes in human welfare, if you can connect. Lactobacillus or lactic acid bacteria, which converts your milk beautifully into gel-like tasty curd. You have your dosas, doklas, again by the process of fermentation using bacteria. Cheese, again by using bacteria and fungi, again your microbes and human welfare. Alcohol, again your brewers' yeast, bakers' yeast, Saccharomyces cerevisiae, if you remember, again using microorganisms. So my dear friends, again microorganisms when we are using to obtain products, even processes, biogas plant, if you remember. So this is nothing but using living organisms to produce products or obtain processes. So this is biotechnology, which is nothing new. Yes, advancement have been made, but this has been continued since ages. So now, what is the new biotechnology, or what is the new field which is such an upcoming field which has a huge panorama of scope of biotechnology? Herein, we are integrating natural science, conglomerating natural sciences and organisms, not just the microorganisms, but also the cells, their parts, or their molecular analogues. Molecular analogues, we basically, when we are talking about molecular analogues, we are talking about the DNA, small parts of DNA, which is, yes, very rightly understood, nothing but your genes. So how are we using genes and integrating it using natural sciences to beautifully obtain products is what is all biotechnology about. So you can see a small part of the DNA, that is the gene, is introduced. So now basically, in a nutshell or in a crux, if I want to explain you, this entire chapter or this entire procedure is this small part. Let us see the overview of it and then we go on to each and every part of it. So we are basically kind of taking a small part of a genetic material, that is DNA. Say for example, if I want to make insulin. Now insulin has a small problem in the form of proinsulin and insulin, which we will be discussing little later. As of now, just remember, we are making insulin, okay? Hypothetically, let us understand we have a gene which directly makes insulin, okay? So we are taking the gene of interest that makes insulin from the genetic material. How are we taking? Are we needing an entire chromosome? No, we are needing a small part of we need a small part of the chromosome, that is our DNA part, okay? So we are using scissors. You cannot be using the same scissors that we are using for cutting up papers or cloth, okay, or threads. We are using molecular scissors because we are dealing with the molecular level. So we are using molecular scissors. With the molecular scissors, which will be, what do you think molecular scissors would be? Anything from outside? Yes, but in what form? Do we actually insert an instrument? No, we are using enzymes which act as molecular scissors. So we are taking molecular scissors. With the help of enzymes, that is molecular scissors, we are cutting a small part. Say for example, we are cutting a small part of the genetic material, that is the DNA. We are obtaining the gene for the insulin. This gene that is for the insulin, we call it as our gene of interest, GOI, or we are also calling it as the alien gene or a foreign DNA molecule. Similarly, we are using a vector. Now, vector is basically a vehicle. Now, say for example, if I want this gene to enter into an E. coli bacteria and produce insulin, can I take this gene directly and insert it into the E. coli bacteria? No, it will not replicate. Why will it not replicate? Because a vector has to have certain characteristic. One of it is ori, origin of replication. That is a part which will help the plasmid, or which will help this particular plasmid to replicate, thereby this gene which we have inserted to express itself. Just putting the gene or the genetic material or the foreign DNA will not express into the host organism, and hence we need a vector. The same way as we need a vehicle to go from your classes to your home or from your home to your NEET center, you need a vehicle. Similarly, for the gene of interest to enter into the E. coli or the host, we need a vehicle, and this vehicle at present, let us say we are talking about a plasmid. We are using the same molecular scissor. We are making a small incision or a cut on this plasmid, and we are taking the gene of interest, inserting it with the help of an enzyme called DNA ligase, which acting as a glue. So we have initially made cuts. Now these cut parts we are joining together. For joining we need a glue. Here we are using a molecular glue which is DNA ligase. So you see our gene of interest is now ligated or now attached on the vector, and this vector has two different genetic materials combined together. Hence we call it as a recombinant DNA. Once this recombinant DNA is formed, this recombinant DNA has to now enter into a host cell. And this host cell, say for example, we are talking about E. coli, we are inserting it into the E. coli. This process of inserting it into the E. coli is the process which is called as transformation. Once transformation takes place, this bacteria, which is now having a recombinant DNA, within the bacteria, this plasmid will replicate depending upon its copy number. Once it replicates, forms multiple copies, it will start to produce the protein that we desired for. That is, it will start to produce insulin. Now, this bacteria will form its progenies. Once it forms its progenies, we call this process as gene transfer. Meaning, now our recombinant DNA is into every progeny of this E. coli bacteria. Gene transfer has taken place. Now, in each of these daughter E. coli or the progeny E. coli, our gene of interest in the form of plasmid or recombinant DNA will start to replicate, will start to produce our desired product, that is insulin, and in a short span of time. Why a short span of time? Because you are well aware again from your botany chapter that E. coli or bacteria will replicate every 20 minutes. So every 20 minutes it will replicate. We will have a huge colonies of bacteria. These huge colonies of bacteria will have huge number of plasmids, and these plasmids are going to produce our product, which is insulin, and we have insulin produced in mass production in a short span of time. Is the beauty of biotechnology. Such beautifully biotechnology has helped mankind in various fields. So this is what we are going to talk about. This is just the basic introduction of biotechnology, of what we are going to talk about in this chapter, wherein we are going to talk about all the various steps. We are going to talk about how to obtain this gene of interest. We are going to talk about how to kind of, make incisions and cuts on the vector. What kind of vector are we going to choose? How we are going to help transform it once transformation takes place. How we are going to obtain it in large production. So let's continue. Now, when we are going to talk about the details of each one of this, let us first go into understanding of what are the two core techniques of biotechnology. When we are talking about the two core techniques of biotechnology, one of the core technique is genetic engineering, the another one is bioprocess engineering. Genetic engineering, our engineer friends, our JEE friends are studying engineering, where they are studying various techniques to obtain different products by certain permutation combination, by certain addition, subtraction, and changing the entire product. Okay, so here also in genetic engineering we are doing that at the genetic level. Techniques to alter the chemistry of the genetic material. Now we know DNA or RNA is a genetic material, mainly it is DNA. So we are taking the DNA, we are altering the genetic material or the chemistry of this DNA, that is the genetic material. We are introducing our gene of interest. So we are taking the DNA, we are introducing our gene of interest, altering the chemistry of the genetic material, and thereby the genotype and thus the phenotype of the host organism. So you introduce these into the host organism and thus change the phenotype of the host organism. But we cannot do this just in the house that we are staying, or just into the room that we have, our study room, or just into the classroom. We need to have a well setup. Why do we need to have a well setup? So that there is no adulteration, there is no contamination, and we have the growth of only those organisms, those microorganisms or those bacteria whom we desire to, and not the other microbes which are present in the environment. So we need to have a sterile, meaning a microbial contamination free ambience in the chemical engineering process that enables the growth of only, please remember this, the desired microbe or eukaryotic cell in large quantities for manufacturing of biotechnological products like antibiotics, vaccines, enzymes, etc. Is this important to remember? Yes, because this can come as our MCQ as to what are the two core techniques of biotechnology or modern biotechnology. Let us quickly go ahead with the question. The question says, bioprocess engineering involves alteration of the chemistry of the genetic material, formation of chimeric DNA in genetic engineering process, isolation of only desired gene for recombinant DNA technology. RDT is recombinant DNA technology. Remember this, they can use this in the paper. Maintenance of sterile ambience in chemical engineering. Giving you 2 minutes. Solve and let me know the answer. So what will be answer? Answer will be yes, maintenance of sterile ambience in the chemical engineering process, without which we cannot proceed further ahead. Now, NCERT says there are 3 main steps of biotechnology. One is identification of the desirable gene. Second is introduction of identified DNA into the host. Third is maintenance of an introduced DNA into the host and transfer it into the progeny or into the next generation. However, this is just a crux. These are all the steps. Identification and isolation of gene of interest. We have to first identify our gene of interest, post which we will have to cut the DNA at a particular specific location with the help of RE, that is restriction endonuclease, which is a molecular scissor, separate and isolated by gel electrophoresis, amplification of a gene of interest using PCR, polymerase chain reaction, ligation of the fragments using vectors, which is and forming recombinant DNA, insertion of recombinant DNA into the host cell of the organism, that is transformation, as I explained you, and then we have downstream processing, and this downstream processing helps us in mass production. So let us see few of the important topics here, such as we will talk about how do we isolate and obtain the gene of interest. Now when we are talking about isolating and obtaining gene of interest, my dear students, our genetic material or gene of interest, if we are talking about, it's a DNA molecule mostly. Now this DNA which is there is present within the nucleus, isn't it? How do we reach within the nucleus? We will have to break through the barriers which are present, such as the cell membrane, go through the cytoplasm, through the nuclear membrane, enter into the nucleus. So what are we going to do is, we are going to have all these broken so that we have our DNA in the pure form, pure from other impurities such as macromolecules, which are RNA, proteins, polysaccharides, lipids, etc. So we are going to first basically break open the cell to reach to the nucleus. Now depending upon what cell are we using for our gene of interest, we will use enzymes accordingly. So if we are using bacteria, the enzyme that we will use is lysozyme. Again in health and disease, if you remember innate immunity, we had spoken about lysozyme as a physiological barrier found in the tears, which acts as antibacterial because it breaks open the cell wall, breaks down the cell wall of the bacteria. If we are obtaining gene of interest from fungus, we are using chitinase. If we are using plant, we are using cellulase, thereby breaking open the cell, breaking through the barrier, entering inside. Now if there is RNA which will be obviously present, we don't need RNA, we don't need protein, we don't need lipid, we don't need polysaccharide. So what do we do is, we use enzyme called RNase. RNase will break down the RNA. We will use protease, which will break down proteins. We will use lipase, which will break down the lipids. We use amylase, which breaks down the polysaccharides. Are we going to use DNase? No, we are not going to use DNase. Why? Because if we are breaking down the DNA, what are we going to obtain? We are going to be left with nothing then. And what will we introduce into the vector? Nothing. So we are not going to use that. Okay. Once we have broken down this, so see, you have DNA with all these unwanted substances which are surrounding it, which are present around it. We need to obtain DNA in the pure form, which we can do with the help of a process called spooling. Now we are going to take all these impurities into a test tube. We are going to add slowly, gradually chilled ethanol into it. Now as we add chilled ethanol, there are a lot of reactions, the ionic changes that take place, because of which our DNA in the suspension will precipitate. Now if you see here, you can see this in the form of a precipitate. So this precipitate, we use an instrument called spool. we use an instrument called spool, and with the help of spool, we will be taking out, or we will be removing a precipitated DNA from this suspension by the process called spooling, where we are going to obtain the DNA, as you can see, in the form of fine thread-like structures. So we are going to use chilled ethanol, which will precipitate our DNA in the form of fine threads. With the help of a spool, we are going to remove it. So we've obtained our DNA. Once we've obtained our DNA, we now have to use restriction enzymes to cut the DNA. Now these restriction enzymes or restriction endonuclease, which is there. Restriction endonuclease are basically the enzymes which are going to cut the DNA from the central phosphodiester bonds and not the peripheral. Hence endo means within, endonuclease. How does this work? When we are putting the DNA, which is the pure form of DNA or the precipitated thread-like structures, on which we are going to add our RE, in the short we write it as RE, we also call it as molecular scissor, and we are going to leave it. What will the RE or the restriction enzyme do? It will inspect the entire length of DNA. Once it inspects the entire length of DNA, it detects specific recognition sequence. Now what are these recognition sequences? These recognition sequences, see, now these REs will not cut anywhere anyhow, then it doesn't serve the purpose. These REs will cut at a specific site, and these sites are called as recognition sequence, which are nothing but basically palindromic sites. What are these palindromic nucleotide sequences? They are basically sequences of 4 to 8 base pairs, which will read the same when you read it forward as well as backward. So say for example, if I am talking about an enzyme, this is RE-ECO-R1. When we are talking about ECO-R1, if we are reading it from 5' to 3', it reads as GAATTC. The same if we are reading it from 5' to 3' on the other strands. My dear children, it will again read GAATTC. So it is reading the same forward as well as backward. You call such sequences as palindromic sequences. REs will identify 4 to 8 such base pairs palindromic sequence and make an incision. If you are talking about ECO-R1, it makes an incision between G and A, thereby producing ends which are sticky, overhanging, staggered or cohesive ends. These are another type of ends. There are one more type of ends which are called as blunt ends. These blunt ends we are not using for the process of biotechnology at our level. For other processes we are using these. So once it identifies its recognition sequence, post which it will bind to the DNA molecule. Once it binds to its recognition sequence, it will cut the DNA at which strand? 5 to 3 or 3 to 5? It will cut at both the strands and break. Break what? Break phosphodiester bonds. This is important. This can come as our MCQ. It's a PYQ which has come in true and false, asking you whether it breaks the single strand or it breaks or it makes a cut on both the strands. So remember, it makes a cut on both the strands, not in the center, slightly away from the center of the palindromic sequence. So it cuts each of the 2 strands of the double helix at specific points in their sugar phosphate backbones, that is phosphodiester bond, it will break and thereby produce, so see the sticky end. So say for example, if you're talking about the foreign DNA, let us first talk about the foreign DNA. So when we are talking about foreign DNA, when we are using ECO-R1, you can see GAATTC, this is a palindromic sequence, means the enzyme that we are using here is ECO-R1, the RE that we are using. It will make a cut between G and A, as I told you. As it makes a cut between G and A, these are kind of sticky ends or overhanging or staggered ends that are formed. Now these ends that are formed, they will have nitrogen bases which are free. We are using the same RE, we are cutting our DNA also you see with the same RE. Why same RE? Why not another RE? Again an assertion reason, because it will produce similar complementary overhanging or sticky ends. So when it forms a similar complementary or sticking end, say for example if you have A here, it will form a bond only with the base pair which, or with the base which has nitrogenous base as T. Again here T, here A, and here A. So if you see T, T, A, A, T, T, A, A. So we need complementary strands and hence use the same RE. It will now join together with the help of an enzyme called ligase, DNA ligase, and form a recombinant DNA. Is this much clear? Very good. Now once we have obtained our gene of interest, this was a little step further ahead that we spoke about because we were talking about how it cuts. Now once we have obtained, we isolated our DNA into small parts in the pure form. Once we've obtained the DNA in the pure form, post which we are cutting it with the help of RE. Now say for example if I'm cutting, I will have multiple small particles, multiple small parts of that DNA. How do I identify which is my gene of interest? How do I separate it out from that? So to separate it out, we use what is called as gel electrophoresis. Now as the name suggests, gel, we are using a gel which is an agarose gel prepared from seaweed. You've studied about gels and seaweeds again in your botany chapter. So we are taking agarose gel which will have a sieving effect. Sieving effect is a similar sieve that you use to strain anything, okay? So it will have small porosity like structure within it. Now we are also applying an electric field. So we have wells on one side which are like depressions where we are going to load our DNA samples. And we have an agarose gel which is present here. We have electric current that will pass through the buffer into which this is placed, wherein on one side we have negatively charged electrode and one side we have positively charged electrode. Now which is the positively charged electrode, cathode or an anode? Anode, positively charged electrode, cathode is negatively charged electrode. Now DNA molecules are negatively charged. So we are placing, say for example if I am drawing here, on this side we have our positively charged anode, we have negatively charged cathode. So we have anode here. Once we have placed our DNA fragments into these wells, we will now pass the current. As we pass the current, now these fragments will start to separate out, as you can see here. So they are starting to separate out. As they start to separate out, since DNA is negatively charged, it will move towards positive charge. As it moves towards positive charge, now who will move faster? The smaller the fragment, the faster and the farther it will move. The larger the segment, the slowly it will move, and it will move very less towards or very slowly towards the anode. So if you see in these wells 1, 2, 3 and 4, 4 has the smallest one which has moved quickly towards the anode. The second, the third have larger fragments which are going slowly, gradually, and this one, my dear children, is not the largest, it is an undigested fragment, okay? Now depending upon, we have already studied our entire genome, we know what will be the size of our gene of interest. Accordingly we have a reference here, and on the basis of that we can now have our gene of interest. But when we are doing this gel electrophoresis, do you think we can see these DNA fragments with our naked eye? No, we cannot. So we will have to introduce a dye called ethidium bromide. When we are introducing the ethidium bromide dye, post which we will expose it to UV radiations. As we expose it to UV radiations, these fragments or these strands will start to appear as bright orange strands. Depending upon our size, as I told you, to separate it, we will kind of cut it out from the gel piece by the process called elution. This can come as our MCQ. Please make a note of it and remember it well. Yeah. So again, This is basically just a gist of it. A sample of DNA, sample that is DNA fragments are loaded in the well towards the cathode. Please remember, under electric field, the DNA will start to separate out. The heavier the fragment, the slower it will move. The lighter the fragment, the faster it will move towards the anode. They are not visible under naked eye, so we will have to stain it with ethidium bromide and visualize under UV rays. It will appear like bright orange strands, and thereby the desired gel piece with the DNA fragment now cut out process called elution. Once we have obtained our gene of interest, we will not directly go and attach it onto the vector. Why? Because we have only one piece of that gene of interest. What if something happens, something goes wrong? What if something, it drops down, it doesn't work, it doesn't bind, it will be a waste, and we'll have to start from the step number 1. So we will have to make multiple copies of it. How do we make multiple copies of it? With the help of a process which is called as polymerase chain reaction, which has 3 steps: denaturation, annealing, and extension. Now, the entire procedure is summarized here in this table. Just give me a second, I'll have a sip of water and then we proceed further. Now, when we are talking about making copies of these DNA strands, we have our original DNA, that is a template DNA, right? Now, the same way as you've studied in molecular biology, in molecular genetics, you've studied that within our body replication takes place, right, of the DNA. Now, the same process taking place outside in vitro, how does it take place? So, this is a double-stranded DNA. We will have to first separate out these 2 strands, so that if this is one of the template strands, I can add one more onto it. If this is one template strand, I can add one more onto it, and we will have 2 copies, 2, then 4, then 8, and then so on and so forth. So for that, we'll have to first separate these 2 strands, isn't it? We know that the 2 double-stranded helical structure is joined together by hydrogen bond, which is a weak bond. To break the weak bond, we can use temperature, we can use acids, we can use alkalis. So for that, what are we going to do? Are we going to use acid, alkalis, or chemicals here? No, we are just going to use a temperature change, which will break this bond. So we put this in a machine, wherein the first step is denaturation. At 95 degrees Celsius, when we are exposing this double-stranded DNA, the hydrogen bond at such a high temperature will break. The 2 strands will separate out. As the 2 strands separate out, we then go on to the procedure called annealing, where we are reducing the temperature to approximately 55 degrees Celsius, and we are adding small short parts of DNA called primers. These are oligonucleotide primers, which are synthesized complementary to the strands. We will now add these to the template DNAs. Once we are adding these to the template DNAs, then comes the function of an enzyme called Taq polymerase. If you remember the very first session of Power Hour where we discussed about enzymes, I had explained you about Taq polymerase, saying that enzymes which are within the body are thermolabile. They will get denatured and damaged at 40 degrees and above Celsius. However, there are enzymes which are obtained from organisms such as Thermus aquaticus bacteria, which are thermostable and can function even at 80-90 degrees Celsius and even higher. So we are putting Taq polymerase enzyme, increasing the temperature to 72 degrees Celsius, and we have already now also added nucleotides ATGC base pairs for different types of nucleotides, and depending upon the complementarity, the Taq polymerase will help, and these will now start to increase the length of the primers, thereby the process called extension, and we now have 2 strands of DNA or 2 double helical DNA, 2 copies of the template DNA. The same procedure we keep on continuing. Our NCRT diagram 9.6, as you can see, and as we keep on increasing, you can see that after particular cycles we can obtain approximately 1 billion of copies of the DNA. Once we have obtained multiple copies of DNA, now we will ligate. The process of ligation, as I explained you, we are having the same RE that we will be using, that is the same restriction endonuclease, which will form complementary sticky ends. They will now be ligated using an enzyme which is DNA ligase. And our rDNA will be formed. Now, when we are forming the rDNA, when we are using a vector, is it that any vector can be used, or will we be using the same vector every time? No, we will be using different vectors such as plasmids, bacteriophage, PBR322, YAC, yeast artificial chromosome, bacterial artificial chromosome, etc. Are there any criteria that a vector should fulfill? Yes. A vector should be capable of replicating itself irrespective of the control of the genome or of the genetic material or the chromosome of the host itself. Second, it should have a high copy number. The number of times that it can replicate within the host is its copy number. The more, the merrier. So the higher, the better. It also should be capable of easily linking to the foreign DNA and should also help. It itself should have the capacity to help us select a recombinant from a non-recombinant, a transformant from a non-transformant. Now, if you are talking about our genetically engineered plasmid, which is PBR322. This PBR322, this is the structure given in the NCERT, where if we go clockwise, you can see there is ori which is present. This ori is origin of replication, which will decide the copy number of the bacteria, of the plasmid, and will also help in replication of the plasmid. Thereby, it will also help in replication of our gene of interest. So, any foreign DNA, when linked to the plasmid should have an ori, without which it is not possible. Also it has to depend upon the size of the vector. The size of the vector dependent upon the size of the host and size of the gene of interest, basically the gene of interest. It should also have cloning sites. Cloning sites are basically special recognition sequences that you can see here, which are for specific RE. Every RE has its own specific recognition sequence. Any vector should have a criteria that it should have more than one cloning sites or more than one RE recognition sequences, so that more than one RE can work. However, for one RE, there should be, as NCERT says, very few or only one recognition sequence. Otherwise, it will form multiple small fragments and complicate the entire process, which we don't want. It should also have something called as selectable markers. Selectable markers are ampicillin resistance and tetracycline resistance. Now, if we are inserting a gene of interest using the enzyme BAMH1 as given in the NCERT, and we have introduced a gene of interest here, ampicillin will help us identify a transformant from a non-transformant, and tetracycline will help us identify a recombinant from a non-recombinant. So this is about the ori that I explained you. I explained you about the cloning sites, where I told you very important. Multiple REs cloning sites, however, one cloning site for one RE. I also explained you that the cloning sites, preferably a single cloning site for the commonly used RE should be present, and it should be in one of the two antibiotic resistant gene regions. Thereby, it will help us identify, as I told you, a recombinant from a non-recombinant and a transformant from a non-transformant. For these selectable markers, we usually use antibiotic resistant genes such as ampicillin resistance, tetracycline or kanamycin, name one and the same thing, or chloramphenicol resistant gene. However, in PBR322, the two antibiotic resistant genes are ampicillin and tetracycline resistant gene. Then we will introduce it into the E. coli. Why are we using E. coli? Because E. coli basically does not have any, it's native plasmid which provides antibiotic resistance. So it is not resistant to any antibiotic. Hence, if we insert a plasmid with the antibiotic resistant gene, it will help us work as a selectable marker. This can again come as your MCQ. Now there's something called as insertional inactivation. Now say for example, if I'm drawing a PBR 322 for you all, you know that this is our gene for ampicillin resistance. This is our gene for tetracycline resistance. We are using the restriction endonuclease, which is BAMH1. BAMH1 we know is in the region of tetracycline resistance. And this is ampicillin resistance. Now, when I am introducing my gene of interest in this region, when I'm introducing my gene of interest, I am kind of breaking the tetracycline resistant gene and introducing or inserting my gene of interest. Now, if this is the gene which is providing tetracycline resistance, if I am cutting it into two parts and if I am introducing, say my hand is the gene of interest, this was originally the tetracycline resistant gene which was providing tetracycline resistance. I am introducing my gene of interest, so I am kind of breaking down this tetracycline resistance gene to introduce my gene of interest. So I am inserting my gene of interest, thereby inactivating the tetracycline resistance gene, because this is now not going to work as it was working. So this tetracycline resistant gene is inactivated because of insertion of my gene of interest, the process called insertional inactivation. Now when I am wanting to differentiate a transformant from a non-transformant, I told you I will be using ampicillin as my base to help identify whether the E. coli has taken up the plasmid or not taken up the plasmid, that is, it has been a transformant or a non-transformant. Once I identify it's a transformant, I will have to then expose it to tetracycline to identify whether it's a recombinant or a non-recombinant. Now this is too much of a tedious process, isn't it? It's a strenuous process. First we grow the colonies, then we put in ampicillin, then we put in tetracycline. Plus at the same time, if I understand the recombinant from a non-recombinant, because of insertional inactivation, my required E. coli with the gene of interest plasmid has died. Once it has died, it is of no use. Though I have taken just a sample from the original colony, but still I have killed the host which had my gene of interest. So to prevent all of this, I will be using chromogenic substrate as a selectable marker. Now, how do we use this chromogenic substrate as a selectable marker? Now say for example, I am taking another vector which is PUC8. Now this name is not given in the NCERT, but it's for your understanding, okay. Now this PUC8, if you see on one side, has a gene which is for resistance against antibiotic, that is ampicillin. And on the other side, if you see, there is a gene called LAC-Z. Now, ampicillin resistant gene does not have any cloning site for any RE. However, if you talk about LAC-Z, it has multiple cloning sites which are present, and for each RE there is only one cloning site. What are we going to do is, now, and it obviously has its own RE. Now so what are we going to do is, using any of the RE, say if we are using the same, that is BAMH1, we are introducing a gene of interest here. So if I am introducing my gene of interest here, I have again inactivated by inserting my gene of interest, the gene of LAC-Z. Now this is insertional inactivation. What does LAC-Z do? LAC-Z basically codes for an enzyme called beta-galactosidase. When you put this beta-galactosidase in a substrate that is X-gal, the beta-galactosidase will break down this X-gal and produce blue-colored products. Now, if I have my gene of interest which is inserted in the region of LAC-Z, LAC-Z is inactivated. If it is inactivated, will it produce beta-galactosidase? No. If it does not produce beta-galactosidase, will it produce blue color colonies? No. Meaning, the bacteria which will have a recombinant DNA will not produce a blue color colony, and the one which has this recombinant DNA will produce a blue color colony, isn't it? So recombinant, no blue color, meaning it will be white color, and a non-recombinant mean it will be blue color. So when I grow the colonies of the bacteria, I will have two different colors. I will have blue color colonies, I will have white color colonies. Blue color colonies means the gene which is LAC-Z for beta-galactosidase is not inactivated because there is no insertion of gene of interest. It's a non-recombinant, means it is of no use to me. So the one that is of use to me is the white colored colonies, because it will not produce blue color because of insertional inactivation of the LAC-Z gene. Hence, by chromogenic substance or substrate, that is color producing substrate, using that I can identify a recombinant from a non-recombinant without killing the host which has my gene of interest. Isn't this exciting? Yeah. So this is how I can use a chromogenic selectable marker. Now similarly, we can also kind of choose the vector not only on the criteria but also on the host. If I want to introduce the gene of interest into an animal cell, my dear children, if I want to produce a rosy cow, which you have studied, I am not going to use a plasmid, isn't it? If I am going to use a bacteria, I am not going to use a retrovirus. If I am going to use a plant, I am not again going to use a retrovirus. But if I am going to use a bacteria as my host, I will be using plasmid. If I am using animal cell as a host, I will be using retrovirus. If I am using plant cell as a host, I will be using TI plasmid. So plasmid we've already studied in detail. This is again the same explanation of the entire thing. If I am going to use the animal cell, I will use a retrovirus, because you know retrovirus has the capacity, as we've studied in HIV, health and disease, that retrovirus has the capacity to incorporate itself into the host cell, isn't it? Similarly, TI plasmid also has the capacity to incorporate itself into the host cell. So these will have the capacity to incorporate itself into the host cell. So if I am putting my gene of interest into these, thereby kind of disarming them, because now I am not allowing them to express, but I want my gene of interest to express. So whatever is the pathogenic part of the gene, I am cutting that out, I am putting my gene of interest there, and thereby disarming the entire pathogen, converting that pathogen into a weapon that I want, hence called disarmed pathogen. I am dropping it off its arms and ammunitions. Okay, so retrovirus and TI plasmid for animal and for plant cell, it will incorporate the gene of interest that I want into the host genome, and the host genome's genotype, thereby the phenotype will change, and it will produce products that I need and that I wanted to produce. Isn't that exciting? Okay. So once I have done that, I will have to now, so I have done that, right? I will have to now introduce it into the host cell by the process called transformation. Will it be very easy? We are just saying that, okay, we are taking our recombinant DNA, we are introducing it into the host cell. Will the host cell easily accept it? The host cell may or may not. So if it doesn't directly accept it, we will have to expose it to certain treatments. So for inserting the recombinant DNA, the host has to be made competent. And And we can do it by different techniques. How can we do it? We can do it with the help of heat shock heat method for bacteria. If we are using it for plants, we can use a biolistic or gene gun. If we are using it for animals, we can use micro injection methods. So these are gene guns for plant cells. These are micro injections for animal cells, with which we can insert our gene of interest into it. The heat cold heat technique is for basically bacteria, not for plant or for animals. Once we have transformed the host, we want the host to grow. We want to culture it. Now initially when we are doing a trial and error method, we will do it on a small scale. When we are doing it on a small scale, we will be doing it in a laboratory, and when we are doing it in a laboratory, we are doing it in petri dishes to mainly obtain the proteins which are our desirable products. So, in small scale production, we are using petri dishes, using agarose gel medium, that is the nutrient medium, and we are incubating it at 37 degrees Celsius, which is the normal temperature, for the bacteria to grow overnight in ovens, and we will have colonies. Once we obtain these colonies, we can obtain the product from it, because these bacteria will now start to produce product. Once we obtain the product, we can do a trial testing method. Once it is seen that trial and testing phase absolutely safe for use, it does not have any harm, it does not have any side effects, it does not have any disastrous effect, we can then do it on a large scale. To produce it on a large scale, we are going to use certain bioreactors which are instruments. So on commercial scale, large scale production, bioreactors are used. These will produce large volumes of cultures, which are approximately 100 to 1000 liters of cultures that can be produced. So basically bioreactors are those vessels in which raw materials are added, biologically converted into enzymes or products using various living organisms such as microbes, plant cell, animal cell or host cell. And to do that, we will have to provide an optimum condition, such as optimum pH, such as optimum temperature, such as a sterile ambience, as you can remember, salts, vitamins, nutrients that are required. Otherwise, how will it flourish, isn't it? There are two types of bioreactors. There is simple stir tank bioreactor and the sparged stir tank bioreactors, where there are stirrers which are there, which will bring about aeration. We've added all the products, we've added all the raw materials, we provided all the nutrients, and it will start to kind of produce products. The bacteria will multiply and the products will be obtained. Here also we are using the same thing, but we are generating bubbles by increasing so that the oxygen transfer increases, the surface area increases, and there is a fast production. Either of them can be utilized. So that is how various products are obtained. Now, where is biotechnology used to a great extent? Biotechnology can be used to a great extent in almost every field. However, commonly used in agriculture and in medicine. So, biotechnology essentially deals with industrial scale production of biopharmaceuticals, biologicals using genetically modified microbes, fungi, plants, animals. You can call them as GMO, genetically modified organisms, GMP, genetically modified plants. So as I told you, we can use it in biopharmaceuticals, in therapeutics, diagnostics, yes, genetically modified crops such as golden rice, we have processed foods, we have bioremediation, waste treatment, we have energy production. Now, when we are talking about these applications of biotechnology, there are basically three critical research areas of biotechnology. Though there is a lot of research happening, these are the three main areas so that things can get better and better. What are they? Let us see. We need to provide a best catalyst in the form of improved organisms, usually a microbe or pure enzyme. We need to create optimum conditions through our engineering for a catalyst to act. We also need downstream processing technologies to purify products and organisms and compounds. So these are three critical techniques or critical fields where we need to further have a research. Let us talk about two points, one in agriculture, one in medicine. When we are talking about agriculture, biotechnology in agriculture, it has helped us a lot. Initially, when the population was less, the amount of crop production which was there was sufficient. As the population started to increase, we've studied in reproductive health also, and we've seen the statistical data. As there was an exponential growth of the population, there was a need to increase the crop production. There was a need so that the crops that are produced are not hampered or damaged by any kind of pests, any kind of organisms that will go and cause damage to it. One of the crops that was helped by biotechnology was cotton. We produce BT cotton, Bacillus thuringiensis, BT. We'll talk about it. Now this cotton plant is a cash crop for India, okay. Now this was, if you can see, this is a beautiful kind of mature cotton ball. This would have been the same, however its fate was different and it was eaten by a pest which was cotton bollworm. Okay. Now this bollworm which is there could cause damage to the crop and thereby reduce the yield of the crop. Now earlier then they found out that there was a particular part of the crop which was not eaten by the bollworm. They did a research and they found out that that part had a bacteria called Bacillus thuringiensis. Bacillus thuringiensis, during its one of the process of its pura life formation, that is sporulation, has a gene. It has a gene in itself, that is cry gene or BT gene. Bacillus thuringiensis BT. Now this cry gene during sporulation would produce a toxin which was in an inactive form called prototoxin, cry prototoxin or cry protein or BT protein. This protein is highly toxic to the insects and acts as an insecticide. Once the insect, that is cotton bollworm which is there, an insect which will now go and eat that particular part of the plant which had that bacillus and had that prototoxin, once it was eaten by cotton bollworm, that would now enter into the gut of the insect. Once it enters into the gut of the insect, the inactive toxin is now activated. Why? Because of the alkaline pH of the gut. It will now, once it gets activated, it will now cause pores in the midgut. Because of pores in the midgut, the cells would swell and would rupture, lysis. If the cells of the gut are only ruptured, whatever the insect is eating is not absorbed. If it's not absorbed, the nutrients don't go to the cells, and once it do not go to the cells, the insect will be killed due to starvation. So scientists found out that this Bacillus thuringiensis bacteria had this gene, that is cry gene. They identified this gene, they isolated this gene and they introduced it into the cotton plant. The BT gene was taken, it was introduced into the cotton plant, and this cotton plant cell, not into the plant as such, but into the plant cell. Once into the plant cell, plant tissue culture was done, which you've studied in botany. Once plant tissue culture is done and the entire sapling and thereby the plant is produced, since we've introduced it at the cellular level, the single cell from which the entire plant has originated via PTC, plant tissue culture, the entire cell, all the cells will have this BT toxin in the form of protoxin, so it does not cause damage to the plant. But when this plant is eaten or tried to be eaten by the bollworm, it will enter into the gut and kill the insect. Thereby the entire plant was itself converted into a pest resistant or a pesticidal plant. Isn't this amazing? And thereby we could prevent the entire plant from being damaged. However, these genes which are there are insect specific. How are these genes insect specific? There are certain genes for certain insects. Like for cotton bollworm, we have cry1AC, cry2AB, and for corn borer, we have cry1AB. So depending upon the target pest, depending upon the crop which needs to be protected, we will be using the choice of gene. This can again come as your MCQ and we need to remember it. There are some strains of insects which are killed by this Bacillus thuringiensis, which is coleopterans, lepidopterans and dipterans. Coleopterans has beetles belonging to it, tobacco budworm, armyworm into lepidopterans, and flies and mosquito into dipterans. Similarly, we can also, as I was telling in the beginning of the chapter about insulin, India is the diabetic capital of the world. As many number of diabetic patients are in India, they are nowhere in the world in that particular country. So, we need insulin for such patients. We can genetically engineer the insulin. How do we genetically engineer the insulin? We take insulin gene, we will use a plasmid vector. With that, we can insert it into the E. coli as an rDNA. Once we insert, we should have the insulin produced. However, it was seen that there was no insulin produced. How come there was no insulin produced? To understand this, let us talk about insulin. The gene that produces insulin in our body is basically for the product proinsulin or the protein proinsulin. Proinsulin has two chains, chain A, chain B, which is joined together by chain C. So you have chain A, chain B, joined together by chain C. Once it is released into the bloodstream, it will now be metabolically changed and the chain C is removed. Chain A and chain B are joined together by disulfide bonds. Where the problem, as I told you earlier in the beginning of the session, was the problem with insulin production was that we needed this insulin in an active form, but the gene was for proinsulin. So how can we tackle that? In 1983, scientists of Eli Lilly, in 1983, scientists of an American company called Eli Lilly said that why not produce chain A separately, chain B separately in the laboratory using E. coli and then join them via disulfide bonds. And that was the beginning of biotechnology with respect to insulin, which has helped multiple diabetic patients to get insulin, because earlier we would take it from slaughtered animals and that would lead to antigenic reactions because it was not human insulin, it was bovine insulin. And hence, with the Eli Lilly scientists helping us produce this insulin, it is a breakthrough and has helped all of us with those who have diabetic with lot of medications. Let us go on to the understanding of a question. C-peptide of human insulin is a part of mature insulin molecule responsible for formation of disulfide bridges removed during maturation of proinsulin to insulin or responsible for its biological activity. So it is removed during the process of proinsulin to insulin and thereby we have only chain A and chain B. So that was all from my side for today. I hope you have understood the topic absolutely thoroughly well. It has been crystal clear. Topics such as spooling, such as gel electrophoresis, such as PCR, etc., etc., BT cotton and insulin not to forget. So next session we will be talking about animal kingdom and structural organization in animals. So I hope sessions are very, very helpful for you all, and the exams are nearing, so prepare well and take these as small last minute revisions which will be very, very helpful. So be healthy, take care, and study well. Okay, so thank you for today.